So, this is next to the last chapter in the course, and it should be fairly straightforward, I'm hoping. We're going to talk about the reproductive systems. So this would be classically the only system in which there is a significant difference between the two sexes. So we will first start talking about the male reproductive system and then the female reproductive system, and including the anatomy and the physiology and how it works. So in the introduction to this chapter, though, in the first several pages, we want to discuss what is the purpose of sexual reproduction. So why did this system have to come about? What does it add to us? So what doesn't it add? Well, most of you realize that it's not vital to your existence, right? People can live their entire lives and not have reproduced themselves and be totally fine, right? And people that have problems with their reproductive system, these are generally not problems that are going to end their lives either. So it's, so this is a good place for us to be. We're really talking finally about something that's, just not as vital, and you may do your entire clinical life or career without really having to counsel people. But it's good for you to know this, and it's good for you to be prepared to do that. And if you have kids, of course, it's really good for you to know this, to be able to discuss it with them on a scientific level. So here, as we look at this, the main purpose, because we always start off, what's the point of all this, is to produce and nurture sex cells, which we also call gametes. And your sex cells in the male are the sperm, and in the female they are oocytes, sometimes known as eggs. All right, and then parts of the system transport them to the sites of fertilization. Now, in the male, of course, fertilization occurs outside of his body. In the female, it's not very far from the site of production in which fertilization would occur. So we want to note that, and then in the next chapter talk about what happens when there actually has been a fertilization event. All right? Well, to get to this point where we can fertilize, there's a process that has to happen in males and females, and this process is called meiosis or meiosis, right? And this has a lot of similar appearances to the process of mitosis that you studied in AP1, but the outcome is radically different, right? So mitosis you did for growth and adding cell numbers, and you did this when you needed to repair or replace cells. Meiosis is really there to produce these sex cells, and then we'll call it something specific, whether it's in a male or a female, but the process in both of them is collectively a process of meiosis. And the reason this has to be done is because fertilization is a union of two separate cells. And so if each of these cells had the normal number of chromosomes that the other cells in your body have, which is 46, well, then the cell produced from the union of these two would have 92 chromosomes, and this would not allow development to proceed. So the process of meiosis must decrease the chromosomal material, right? So that each sperm and oocytes have half of what your normal cells have, which would be 23 in sperm and 23 in oocytes. So then when they fuse at the point of fertilization, you would get a cell with 46, which is the right number, right? Also to this, we add that we want some variety. We want our kids not to look like exact hybrids or exact copies of us. We want them to have a lot of different good, bad, indifferent characteristics, but variety. And because variety allows for choice, and choice, of course, being natural selection in the process of evolution. So gene diversity, variety, is a really good thing for the natural progression of our species, the human beings. All right, so here we have, very similar to the sagittal board model in lab, the image of the male reproductive system. All right, so it's the meiotic center, right, which is a testes, so this is where meiosis is going to go on, and then a bunch of tubes, right, that just simply move that sperm. So produced here and then moved into, right, the epididymis and the vas deferens or ductus deferens, and then around to the ejaculatory duct, into the prostatic urethra and the intermediate urethra or membranous urethra, urethra, and the penile or spongy urethra, and out. All right. So the testes is the main site, and so we look at this. This is housed in the scrotum, right, which is probably not news to you guys. So the scrotal sacs, all right, and here we're looking at it from like a frontal view of the scrotal sac. Here in unopened, here as we cut into this to try to look, we notice that the testes eventually, they're enclosed in several layers of muscle. There's a dartos on the outside, and then surrounding there is a cremaster muscle. And they are able to, right, bring the testis closer to the body cavity or further away from it. Okay? So they elevate the testes, bring them up, or they let them go further away. All right? And this is going to be important for the production of sperm, which must be about 2 to 5 degrees Celsius cooler than the overall body temperature. So body temperature is somewhere around 37 degrees Celsius. Sperm production is generally somewhere around 35, right? So it's 33 to 35 degrees Celsius. And so if sperm gets too warm, right, it won't form correctly, and if it doesn't form correctly, it can't fertilize. So warm sperm mean you're infertile, right, as a male. So when we cut in through all of these, we see the testes are enclosed with the epididymis in these layers of connective tissue, all right, and then they leave the scrotal sac through the ductus or vas deferens, which is part of what we call the spermatic cord, all right. So again, that's your overall appearance of this area, and we're going to look more specifically at the testis to see how this works. So if we again cut into, right, the scrotum and take a closer look at the parts of the testis here and the parts of the epididymis, right? What is the role of the testis? Well, it's going to, of course, produce sperm through this process of meiosis, and we're going to give it a special name since it's sperm that's the result. And it's also going to produce the major androgens for the male, and the primary one is testosterone. That would be made here in the testis as well. So the testis, we're going to take a closer look at these features that we call the seminiferous tubules. All right, and they are congregated in these structures called lobules. And in these lobules, again, there would be several of the actual tubules, and there's roughly about maybe 250 or so lobules, or actually 300 lobules here, as I see down here. And so there are thousands of these seminiferous tubules. All right. So if we look at these tubules, we focus in on them, we notice that they are layers of spermatogenic cells, so sperm-producing cells, and then there's these sustentacular cells. So these sustain. These are the nurturing cells. They help provide nutrients to the sperm and help guide the sperm, basically, functional, so they can become functional. And then also inside the testis are these interstitial cells. So they're outside the actual tubule, but within the testis, and they're also called the Leydig cells, or again interstitial cells, and these are going to be the sites where testosterone is made. All right, the tunica albuginea is this connective tissue that is right around this, and it helps to create the actual lobules. And then there's an outer, right, tunica vaginalis that we also see often in the scrotal sac that you see right here. It surrounds everything. All right, so if we look at the seminiferous tubules up close, highly magnified, here's one tubule of the many, thousands in here. So here again, that's the wall of the tubule. This dark line is a basement membrane, so it's the boundary. Okay, so outside the tubule, and here's another tubule right next to it. So in these spaces outside the tubules would be the cells of Leydig, or the interstitial cells. All right, just to let you know that. And these cells would again make, right here it shows them pointed out here, they would make testosterone. Right, these are actually sperm forming cells, and then every once in a while and I don't point these out in lab, but there'll be these really tall, right, Sertoli or sustentacular cells. All right, so what this is layers of sperm cells in various states of meiosis. So here closest to the boundary, this dark line, are the most immature cells that are called the spermatogonia, and then they start by just undergoing mitosis to replace themselves and then produce a cell that will start into meiosis. All right. Now, also I want to caveat, meiosis is not going to begin until puberty in males, right? And we're going to outline why that occurs. But so starting at puberty, these cells would begin, right, producing 2 daughter cells. Then one of those daughter cells would become a primary spermatocyte. It would be in meiosis 1, right? And then it would continue into, there's 2 phases of meiosis, so it would also go into a meiosis 2 as it got closer in. And at that point we call it a secondary, right, spermatocyte. And then it would undergo some changes so that it, right, from here, most immature, change it going through meiosis in here, and then some additional changes here to then have sperm-looking cells, or spermatotids, right, that would be released here. So, again, you'd also see these sustentacular cells. These are very large cells that help develop the sperm, provide nutrition to them, and guide them, and chemicals to help them. They do help with the blood-testis barrier, so this protects the sperm from your immune cells in the body. Again, the sperm come in different stages, so most immature, primary spermatocyte, secondary spermatocyte, spermatids, and then sperm is finally what is actually released here. Alright, so this is looking at this process of meiosis that we specifically call, right, spermatogenesis. And spermatogenesis is going to involve meiosis, and it's going to involve, right, this what we call spermiogenesis. So this is a step in the process, collectively known as spermatogenesis. As I say here, it begins at puberty, and we go from the spermatogonia to the sperm in about 64 days. That's the length it takes, and it depends on these, right, sustentacular or Sertoli cells helping it. And so this cartoon is just showing you, here's our spermatogonium here and here. The 2 end just refers to the 46 chromosomes, 2 pairs of 23. And they undergo mitosis, forming a primary spermatocyte, which then enters meiosis 1. When it completes it, you have 2 secondary spermatocytes. Each of these go into meiosis 2, and so you get these 4 spermatids. The spermatids then undergo some morphological or shape changes, right, from roundish cells to finally these spermy-looking cells that we do call sperm or spermatozoa. All right. I said, again, and this just elaborates on this process, spermiogenesis. So I say meiosis will get the chromosome number correct, which should be 23, but the structure or morphology is in the process of spermiogenesis. So the spermatids reduce their cytoplasm, form a flagellum, and again, they become very small and very streamlined. They develop a special organelle called the acrosome, right? And then it is completely surrounded by a plasma membrane, nucleus. There are centrioles that then help to move this tail that we call the flagellum, and that movement that they do, so they're turning it, is powered by the mitochondria here. All right. So there are 3 parts to the sperm that you can see on your board models: the head with the nucleus, and acrosomes with a special enzyme called hyaluronidase. And then the mid piece here has a lot of mitochondria, and then the tail is known as the flagellum. All right, sperm transport, all right, is through these tubes. So after we get the sperm, the chromosome number correct and the shape correct, then we're just really moving them on. All right, and sperm are constantly forming in the male, and as such, they're always getting pushed through the tubes. So there are 2 epididymids, so they're paired again, epididymides, and so singular epididymis, and these are highly coiled within the scrotum, right, but outside the testis, and they help the maturation of the sperm. So it generally takes about 12 days to move through. They're a place where they're going to store, sperm is going to be stored, and it's going to basically become able to move. So these are actual images from your lab slides to show, close up on the walls. So these are epididymis from 2 separate species. Here's these pseudostratified cilia, right, stereocilia. That's what the wall is made up of. And this one looks more like a columnar, actual simple columnar, but pseudostratified in 1 layer, and then cilia, right? And we often see sperm in the lumen of the epididymis. We will pick up with the rest of the male on the next video.